Okay, so in this video, we're going to talk about 10.3 and 10.4. So we're going to get into the really nitty-gritty of essentially definitions. We're going to talk about muscle contractions and muscle relaxation. So we're going to start off here with 10.3, which is skeletal muscle fibers as electrically excitable cells. This is going to be kind of like the preface to 10.4, which is really on this exam what I want you to focus on 10.4. All right, so again, you have to know this for all of this stuff for both this chapter leading into 10.4 and for when you get into A&P 2 for cardiac muscle. This will be really, really helpful if you understand this. All right, so membrane potentials definition here are due to an unequal distribution of positive and negative ions, cations and anions, across a plasma membrane, which results in a polarized resting state. So essentially a charge difference. So the negatively charged ions exist in the cytosol inside the cell. Positively charged ions exist on the outside of the cell. So this separation of charges creates an electrical gradient that represents a source of potential energy. So I don't really want to get too much into physics here, but we'll talk a little bit about it. So when the barrier separating ions is removed, they're going to follow their gradients. Again, it's like if you shoved 1,000 people into one little bitty room, and if you opened the door, they're just going to kind of like fall out, if not run out. They're going to follow their gradients, okay? So this creates a flow of electrical charges, and the potential energy becomes kinetic energy. So this is why the electrical gradient can be referred to as an electrical potential. All right, so away from the sarcolemma, positive and negative ions are present in about equal numbers. So this means that the cytosol and the extracellular fluid are always electrically neutral. Okay. The membrane potential of a cell is the electrical potential or charge difference that exists across the plasma membrane. A difference in charge or potential between 2 points is called a voltage. The potential across the sarcolemma is quite small and is therefore measured in a unit called a millivolt, or 1/1000th of a volt. So the resting membrane potential is the electrical potential across the sarcolemma of a resting muscle fiber, and it measures right around negative 580 millivolts. So what does this really mean? What's the gist of this whole slide? Essentially, the cytosol is 85 millivolts more negative than the extracellular fluid. So it's more negative on the inside of the cell versus the outside of the cell. And again, the unit is around negative 85 millivolts. It's important to know. All right, so recall that the phospholipid bilayer of any plasma membrane is impermeable to ions. So what that means is those positive and negative charges can't go in or out without special permissions. So the resting membrane potentials change only when the barrier to ion movement is removed or opened from the plasma membrane. So the sodium and potassium ions can move through the sarcolemma using, again, protein channels or carriers. They have to have a door to move through. But they will only move by diffusion if a gradient exists between two regions, so more of something on one side versus another. That's what that gradient means. More something on one side versus another. So this concentration gradient is maintained by the sodium-potassium pump. So if you remember the sodium-potassium pump, that was way back in chapter 3. We talked about this, but I'm going to kind of summarize here. So the pump moves 3 sodium ions out and 2 potassium ions into the cell per ATP hydrolyzed, per energy unit used. So ATP hydrolysis is necessary because this pump moves ions against their concentration gradients. So again, why do we need energy to do this? Well, it's kind of like shoving you. Let's go back to the room analogy. So we have a small room and we have 1,000 people in that room. So instead of opening the door and letting people fall out, we open the door, but we shove people inside and keep them in the room, and then we see a random person on the street. Oh my gosh, there's another person. There's another one for my collection. And you grab them and you shove them in the room and then close the door. It takes a lot of energy to keep those people in those room and get that other person that doesn't really want to go off the street into that room. Sorry, that makes me sound like a crazy person. I'm really not. That's a good example though. So why is ATP hydrolysis, why is energy needed? Because the pump moves ions against their concentration gradients. Ions are like those people. So this creates a high concentration of sodium in the extracellular fluid outside, while the concentration on the inside is low. So this creates a high concentration of potassium inside the cell, when the concentration in the outside of the cell is low. So the sarcolemma of a skeletal muscle fiber has millions of these pumps, ATP pumps, I meant sodium potassium pumps, which work constantly to maintain that concentration gradient, both of sodium and potassium. So the gradients are critical because they provide the driving force for the passive diffusion of sodium and potassium ions through the membrane channels, which we're going to see when we talk about muscle contractions. These are absolutely essential. Okay, so these last 3 slides again are just explaining how polarity constantly exists, how those charge differences constantly exist across the plasma membrane. So that's all just trying to maintain for what we're about to discuss. We're just maintaining that gradient for what we're about to talk about. All right, more definitions here, but you do need to know this definition. So action potentials are brief changes in the membrane potential of a cell from a resting negative value to a positive value, and then back to its resting negative value. So these changes can be used to electrically communicate with and sometimes stimulate a response from other cells. So essentially what we're going to do here is go from negative 85 millivolts, our resting membrane potential, all the way up to plus 30, and then we're going to go all the way back down to negative 85. That's essentially what we're going to do. That's what it's talking about here. All right, so gated channels contain gates, like a door, that are normally closed and open only in response to some kind of a stimulus. You have to have a very specific key to open this. So action potentials are generated by opening 2 types of gated ion channels in the plasma membrane. 2 different types. You can have ligand-gated channels, which are chemically gated, or voltage-gated channels, which are electrically gated. So ligand-gated channels open in response to the presence of a chemical or ligand. Again, all this is saying there's a certain kind of chemical key that it takes to open this door. So what happens is the chemicals are released and they're going to come down and bind, and that lets in sodium or potassium or whatever it may be. That's a ligand-gated channel. Versus voltage-gated channels open in response to action potentials or electricity. So they're open in response to changes in the membrane potential of the plasma membrane. So all of a sudden we go from negative 85 millivolts to plus 30 millivolts. Maybe this channel opens, which that would be actually potassium, but in that particular instance. But let's say we're moving from certain voltage to another voltage. That's going to allow these sodium channels to open, and sodium is going to go in. All right, so let's talk about action potentials. What's the whole process of this? So we'll start at the polarized or resting state, which is again negative 85 millivolts inside and positive outside. So the action potential occurs in 2 stages, depolarization and repolarization, which we're going to talk about. So depolarization begins when voltage-gated sodium channels open. This allows sodium to flow inward. The entry of these positively charged sodium ions, cations, makes the membrane potential become less negative, less polarized, hence the term depolarization. So if something is polar, there's a charge difference. So if you're depolarizing, you're making less of a charge difference. So these sodium ions are coming in, and the inside is, instead of being negative, it's going more and more and more and more and more positive. So less negative, less polarized. So the membrane potential quickly reaches all the way from negative 85 millivolts to 0 millivolts, and then all the way up to plus 30 millivolts. That's really, really positive on the inside of the cell. So something happens when we reach plus 30 millivolts. This is going to trigger repolarization, not depolarization, repolarization. So repolarization begins after sodium channels have closed. Again, at plus 30 millivolts, what happens is the sodium channels snap closed. And at the same time, the voltage-gated potassium channels snap open. This allows potassium to diffuse out of the cell. So potassium is positively charged. If you have positive ions leaving the cell, this means you become more negative. So the loss of positively charged potassium ions makes the membrane potential more negative, returning the sarcolemma to that resting potential, that negative 85 millivolts. So the potassium channels will close once the cell returns to its resting membrane potential, negative 85 millivolts. And we're there to start the process all over again. All right, so we talked about, I think it was 10.1, conductivity. Remember, there was a whole list of definitions, and I said, you need to know these. So recall that a property of muscle fibers is conductivity, which means, again, that electrical changes across the sarcolemma are not isolated events. Action potentials don't stay in one place. Rather, they're conducted, or the term propagated, you'll see that again, propagated throughout the entire sarcolemma. So think about it like ripples on the surface of a pond, flowing from one to the next, to the next, to the next, flowing further and further away from that original source. So this process is very fast and results in the depolarization of the entire sarcolemma, including the T-tubules. We'll see why that's important in a little bit. So the arrival of action potential at the T-tubules is what actually initiates a muscle contraction. And again, we'll see this in just a minute. Okay, so if that whole thing was really wordy for you, you really like pictures, this is a really good summary overview. Action potentials. So again, action potentials are divided into two main phases, the depolarization stage and the repolarization stage. I'm going to kind of summarize this just one more time. So in response to a stimulus, voltage-gated sodium channels open, and the sodium enters the cell. So positively charged entering the cell, this makes the membrane potential less negative. Okay, so we're going from negative 85 millivolts. This continues happening until we make it all the way up to plus 30 millivolts. Okay, so what happens at plus 30 millivolts? Repolarization. This is our next stage, repolarization stage. So we hit plus 30 millivolts. These are going to, bam, snap shut. And these are going to, bam, open up. So potassium is at a real high concentration here versus here, inside the cell versus outside. So what's it going to do? It's going to follow its gradient. It's going to go outside the cell. Okay, so potassium is also plus charge. It's a cation. So what's it going to do? It's going to leave the cell. When you have positive ions leaving the cell, you're going to become more and more negative. So potassium ions leave the cell, making the membrane potential more negative again, all the way down to negative 85 millivolts. Okay. All right, let's talk about the process. This is the main point here. The process of skeletal muscle contraction and relaxation. You'll need to know this for an exam. All right, so all skeletal muscles are what you call innervated. A single neuron called a motor neuron. Just think of it as a neuron. You don't have to really think about motor neuron or whatever. Just, it's a neuron. It's going to communicate with potentially many muscle fibers. So each connection, neuron and muscle, is referred to as a synapse. So the neuromuscular junction, or NMJ, is a synapse where a motor neuron communicates with many muscle fibers. So the function of the neuromuscular junction is to transmit a signal called a nerve impulse or an action potential, we've heard that before, from the neuron to the sarcolemma of the muscle fiber. So basically communicating between nervous system and muscles. Okay. So this is what your neuromuscular junction would look like. Again, it's essentially the connection between and communication between a neuron and a muscle. Okay, so the axon terminal of the neuron contains synaptic vesicles filled with acetylcholine, abbreviated ACH, which is a neurotransmitter. So the axon terminal is essentially this ending portion of the neuron. The synaptic vesicles are just, I used to think of them as like little bubbles little bubbles filled with chemicals. These are your synaptic vesicles here. They're at the very, very end portion. Okay. And those little bubbles, those synaptic vesicles, are filled with acetylcholine. It's a neurotransmitter. It's a specific one. And essentially, a neurotransmitter is just a chemical. So really, our goal, our function, our purpose here is to communicate between the nervous system and our muscles, depending on however many it is. But how do we do that? We do that with chemicals, and we do that with specific chemicals called acetylcholine. Okay, that's a neurotransmitter, and we're going to talk about how that happens in just a second. I'm getting a little ahead of myself. All right, so neurotransmitters, again, are chemicals that trigger changes in the target tissue when released. This allows for, again, one of our core principles of A&P, cell-to-cell communication. So the synaptic cleft is a space between the axon terminal and the muscle fiber. So this would be your synaptic cleft here. Space between this and this. Between muscle and neuron. Synaptic cleft here. So the synaptic cleft is also filled with collagen fibers and a gel that essentially keep everything in place. Okay, so the motor end plate is a specialized region of the muscle fibroplasma membrane. This is actually, so this is our neuron here. This is our muscle. This is specific. This is your motor end plate here. This is a specific part of the plasma membrane of your muscle. Okay. It has a really folded surface, which on that surface you have lots and lots and lots of ligand-gated sodium channels. So again, what's a ligand-gated channel? Again, basically you have to have a certain kind of chemical bind and it'll open the channel. And these are in particular sodium channels. So what do they let in? Sodium. Makes sense. So again, ACH is abbreviation for acetylcholine. Acetylcholine is the ligand or chemical that opens up these gates. So when acetylcholine binds to these ligand-gated sodium channels, this allows sodium to diffuse into the muscle cell. Okay, makes sense so far. All right, so muscle contraction can be broken down into 3 phases. The excitation phase, the excitation contraction coupling, and the contraction phase. Okay. So the excitation phase begins when an action potential signals the release of acetylcholine from the axon terminal into the synaptic cleft. Essentially, it's your brain telling your muscle you need to move. The excitation contraction coupling is a link between the stimulus and the contraction. The contraction phase begins when calcium ions bind to troponin, which pulls tropomyosin away from the actin's active site, and the cross-bridge cycle begins. This is actually when we get to our muscle contraction. All right, so we're going to talk about each phase, and I have specific videos on each of them, so if nothing I'm saying makes sense, you can watch those videos and you can actually watch like an animation. I love the animations. I'm kind of like a visual person. So the animations really helped me. So I'll try to go through them, but I'm not going to actually show you the animations on here. That's for y'all to watch if you want to watch that. All right, so hopefully it'll help you. So the excitation phase, which is our first phase of muscle contraction, is essentially when you have an action potential from the brain or the spinal cord, the central nervous system, that's going to arrive at the synaptic terminus of the motor neuron. So this signals the release of acetylcholine from the axon terminal into the synaptic cleft. So acetylcholine diffuses across the synaptic cleft and will bind those ligand-gated channels found in the motor end plate of the muscle fiber sarcolemma. The ligand-gated channels open when they bind acetylcholine, which allows sodium ions to enter the muscle fiber, generating an end plate potential. Okay, that was really wordy. Let me show you on this picture, okay? So basically what's happening is skeletal muscle is voluntarily controlled, right? So we need our brain to tell our muscle, you need to move. And this is how the brain does that, which we go over a little bit in the next few chapters. But for right now, we're talking about when that signal gets to the muscle. So we have essentially an action potential coming down here from the brain to the very end of our neuron, the axon terminal. So at the axon terminal, you have these little bubbles, these little vesicles, synaptic vesicles, which are filled with chemicals. That chemical is essentially a neurotransmitter. It's a specific one, It's acetylcholine. So a little hint to you, if you take a test, whether that be a nursing test or a test in my class or A&P2, and it's talking about a neurotransmitter and you just have no idea what possible neurotransmitter it could be, you're given a list, acetylcholine is one of those. 99.9% of the time, acetylcholine is going to be the right answer. Just so you know. Little side note. All right, so we have the action potential arriving at the axon terminal. So once it arrives at the axon terminal, those synaptic vesicles are going to be prompted to release. They're going to bind down here, and they're going to release all their nice little granules here. These little blue granules. So again, these little blue granules are a neurotransmitter called acetylcholine. So why are we doing this? What purpose does this serve? Well, we're trying to communicate between nervous system and muscles. So how do we do that with this acetylcholine? Well, it goes down here, and these are ligand-gated channels. They need a specific chemical in order to open, and this just happens to be the right chemical, acetylcholine. It's going to bind, fits perfectly, and when it does that, sodium is going to flow in. So the entry of this sodium will depolarize. So again, depolarize. There's not that charge difference anymore. We were negative in here and positive out here, but now we got positive sodium coming in. Now we're positive in here. So it depolarizes the sarcolemma locally, leading to an end-plate potential. Okay, so again, an end-plate potential is essentially depolarization at the end plate. So this, again, occurs as a result of sodium ion influx. So multiple end-plate potentials typically have to be generated in order to stimulate an eventual muscle contraction. So motor neurons continue to fire action potentials. Your brain keeps signaling, contract these muscles, as acetylcholine is rapidly degraded by the enzyme acetylcholinesterase, which is present in the synaptic cleft. We're going to see acetylcholinesterase later when we talk about muscle relaxation. It's going to be playing a big role. So what it's saying is, in this little cleft here, you not only have these lovely little granules, but you have these little enzymes, acetylcholinesterase, which is specific for this neurotransmitter. What it does is it makes it not really able to bind here anymore. It makes it not really functional anymore. So that's going to play an important role when we get to muscle relaxation. Okay, so that was our first phase. We're going to talk about our second portion here. So excitation contraction coupling is the link between the events at the neuromuscular junction and the actual contraction. So an end-plate potential leads to the opening of not ligand-gated but voltage-gated sodium channels in the sarcolemma surrounding the motor end plate. This is going to trigger an action potential. So again, action potentials propagate. They flow just like ripples in the water. Depolarization of one area of the membrane triggers the next and the next and the next. And this process continues like a chain reaction down the muscle fiber to the triads. So the action potential signals the terminal cisterna to open voltage-gated, not sodium, not potassium, it's calcium. Voltage-gated calcium channels. This releases calcium into the cytosol. Things are getting interesting here. All right, so again, this action potential ripples all the way down and eventually gets to our triads here, where these calcium channels will open, calcium exits those terminal cisternae, and that leads us to our next part. So this video here, I'm not going to actually show it to you. That shows you everything I've just talked about so far. It's really good, really detailed, because it actually moves, it actually shows you what's going on. I really like it. Again, if you're kind of like a visual learner, these are really helpful, and I put them on each section, so it hopefully kind of breaks it down for you. All right, so we get to the best part here. Muscle contraction. Woo! We're excited. We're so excited. We're ready to do this contraction. So what happens here? So calcium ions are released again from that terminal cisternae, and they're going to go down and bind to this lovely troponin. So troponin has 3 subunits. One will bind calcium, one binds actin, the other binds tropomyosin. That's not really important. What I want you to know is calcium will bind to troponin. That's what I want you to know. So when calcium binds to troponin, that causes the little blue protein, little blue regulatory protein, tropomyosin, to move. Conformation change. And the active sites of actin, the little purple beads, they're going to be exposed now. Okay? All right. So this is what this is showing you here. So calcium has been released. And again, at rest, when we don't have a muscle contraction, this blue protein, this tropomyosin, is going to be covering actin's active site. So what happens when we release all this calcium, it's going to come down and it's going to bind onto troponin. Okay, troponin. When calcium binds to troponin, the blue portion here, tropomyosin, will move. It shifts over. Shifts over. Like my sound effects here. And then, now that that's moved, these active sites on the pearls, actin, can bind to myosin. Greatest love story ever. So here's another little animation, just preparation for contractions. Everything I've just told you, only without the cool sound effects. All right, so the contraction phase begins when the actin's active site is exposed, initiating the cross-bridge cycle. So the myosin head becomes cocked once ATP provides the energy. Once cocked into its high energy position, or ready to work position, with ADP and P remaining attached to the myosin head, the head is able to bind to the active site of actin. So note that the cross bridge is at a 90-degree angle relative to the thick filament. And as an adenosine diphosphate and phosphate, that's what that is, sorry. All right, so a power stroke occurs when ADP and P are released from the myosin head. So myosin pulls the actin towards the M line as it pivots it to its relaxed or low energy position. The cross bridge is now at about a 45-degree angle relative to the thick filament. So myosin can bind another ATP, which breaks the link with the actin active site. The attachment of the myosin head does not allow the thin filaments to slide backward, however, because at any given time, some of the myosin heads will still be attached to the actin. So the cross-bridge cycle may be repeated as long as there's two things. You have to have a stimulus to contract and energy available, ATP available to do that. All right, so this is just everything I've said, only in a pictorial form. So ATP, hydrolysis, converting from ATP to ADP, essentially cocks that myosin head, going from here to here, so we can bind. All right, so the myosin head will bind to actin, and then from there, the power stroke occurs when ADP and phosphate detach, or ADP and P detach from the myosin head, and myosin will pull the actin towards the centers of the sarcomere. So the ATP binds again and breaks that attachment in order to do all of this over and 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 over again until we run out of energy or until we run out of your brain saying to do this. All right, so this is actually a pretty cool animation, and it actually shows you very, very, very well in a pictorial form of what I've just said. Hopefully this will help you. All right, so again, the contraction cycle is repeated over and over again. So the myosin head is recocked and it binds to the first actin molecule, and the power stroke repeats. So just saying, power stroke is like one of my favorite terms in all of A&P. Just a cool term. So myosin then binds to the second actin, and so on over and over. For an average contraction, this process will repeat about 20 to 40 times for each myosin head in each sarcomere of the muscle fiber. Okay. All right, let's go to this last little portion here. Let's talk about botulism and Botox. So the bacterium Clostridium botulinum produces the most lethal known biological poison. As little as 1 gram of crystalline toxin is enough to kill about 1 million adults. Think about that. That's not very much. Not very much of this toxin. So exposure to the that's supposed to be botulinum, not botulinum, botulinum toxin through contaminated food causes the disease botulism. So the toxin binds to motor neurons of the neuromuscular junction and blocks the release of acetylcholine from synaptic vesicles. This in turn paralyzes the affected muscle, and without proper treatment, death from respiratory failure will follow because, again, your diaphragm is your major muscle in breathing, respiration, and that is a skeletal muscle which works on this system, so respiratory failure will follow. So this toxin can be used to treat painful muscle spasms and migraine headaches when injected in minute quantities. Also, it is typically used cosmetically to relax facial muscles in the form of Botox. This is actually botulism or botulinum toxin. Kind of cool. All right, so we talked about all the multiple steps of muscle contraction. Let's talk about muscle relaxation. Muscle relaxation, when compared to muscle contraction, is a whole lot simpler. So muscle relaxation has two components. So motor neuron action potential will stop signaling for the release of acetylcholine from axon terminals, and calcium ions are pumped back into the sarcoplasmic reticulum terminal cisternae. All right, so there's a specific sequence of muscle relaxation. So remember that enzyme in that synaptic cleft. So acetylcholine esterase degrades the remaining acetylcholine, again, ACH is our abbreviation for acetylcholine, so that the ligand-gated sodium channels close. The end plate potential ends and the final repolarization begins. This elicits that stop in signaling. Okay. We stop the signaling, and any of that's left, any acetylcholine that's left, will be degraded. So this elicits our no more signaling for this. The sarcolemma also returns to its resting membrane potential as a result of potassium efflux exiting through voltage-gated potassium channels, and calcium ion channels in the sarcoplasmic reticulum close as the T tubules repolarize. So calcium ions are also pumped back into the sarcoplasmic reticulum. This returns the calcium ion concentration in the cytosol to its resting level. So basically, we're pumping our calcium ions back where they came from. In the absence of calcium, troponin and tropomyosin will block the active sites of actin, and the muscle relaxes. So myofilaments will slide back to their original positions, and this is with help from actin and other structural proteins as well. All right, so this one is kind of like an overview of relaxation. All right, talk for just a second about rigor mortis, and then we'll be done for right now. This was a long recording. All right, so rigor mortis. This is a progressive stiffening or contraction of skeletal muscles, which begins about 3 to 4 hours after death. So what happens with rigor mortis, essentially pumps that drive calcium ions back into the sarcoplasmic reticulum, they don't have any energy, so they can't do it. They can't pump calcium ions back into the sarcoplasmic reticulum. So as a result, calcium ions remain in the cytosol, and they're going to bind to troponin and initiate muscular contraction all over the body. So the muscle fibers, they're essentially unable to relax without energy to do so. So the myosin heads can't detach from actin. So the muscles remain contracted until essentially the proteins of the myofilaments fall apart. They degenerate, which is about 48 to 72 hours after death. So it's actually kind of cool to think about the chemical process of all this, even after death. Really cool.